In this video, we will discuss how this lectin-glycan interaction can impact the immune response, and this lectin-glycan interaction will also be affected by the maturation or the type of glycosylation that has occurred on the receptors of the either receptors of the immune cells, such as the T cells, B cells, or the regulatory T cells. So we will start with these T cells. In the T cells, the apoptosis of the deactivation or apoptosis of the T cells can be triggered either by interacting of these galactins with these glycoreceptors such as CD45, CD43, CD7, CD29, TAM3, and this is the interactions is depend on the type of glycosylation that has occurred on the galactins that are expressed on the T cells. So that will trigger apoptosis or this upregulation of interleukin 10 and downregulation of interferon gamma, and these things can also be triggered by these receptors, the C-type lectin interactions, such as with the help of these receptors such as CD43, CD45 T cell receptors, CD43 with this MGL, MHC, Select1 respectively of these antigen presenting cells. So when either of these interactions occur, it will trigger apoptosis of these T cells. However, when these galactins that are expressed on the T cells interacted with the galactin receptors that are expressed on the T cells interacted with this galactin 4, it will lead to the activation and upregulation of this interleukin 6. And these interactions are all based on the type of glycosylation that has occurred on these receptors of the T cells. Based on the different glycosylation, different kind of interactions can also occur, and the other kind of interactions is shown on this bottom of this figure, where what will happen is this: the interactions with the help of this DG signs and ICAM3, and this will trigger this this will trigger uh, uh, immunological synapses, in which what will happen is that the galactin 1 interactions will lead to the upregulation of this interleukin 10, and also it will lead to the proliferation of this regulatory T cells, and these things will occur when this immunological synapses and this DG signs and ICAM3 interactions, both of this occurs. When this is both of these occurs, then it will lead to this regulatory T cells upregulations and interleukin 10 upregulations. However, when this portion will occur when this in the immunological synapses, this galactin 1 interactions will occur. However, when these galactin 9 interactions will occur, galactin 1 and galactin 9 interactions will occur with this Th1, Th2, Th17 cells, it will lead to different kind of signaling pathway, and based on the glycosylation of this T cells. So what will happen with Th1 cells and Th17 cells? It will trigger apoptosis when it interacts with the antigen presenting cells. And however, the Th2 cells are resistant to this galactin 1 and galactin 9 binding, so it will not interact with this galactin 1 and galactin 9 that are present in the extracellular matrix. So these conditions will lead to the selection of this Th2 cells and proliferation of this Th2 cells, whereas Th1 cells and Th17 cells will be died, or because of the apoptosis that is triggered when it interacted with the antigen presenting cells. And similar lectin glycan interactions will also affect these B cells fate. For example, this CD43 that are present on this antigen presenting cells interacted with this Siglec, because if the CD43 has more sialic acids that has the affinity towards this Siglec1, Siglec2, and so what will happens when the Silex interacting with this antigen presenting CD43 that are expressed on the antigen presenting cells? It will lead to this proliferation and survival of these B cells, and these things will also this proliferation survival will also occur when this galactin 1 and select to interact with the B cell receptors that are expressed on the B cells, B cell surface. So there are different ways that might lead to the same outcome, that is proliferation and survival. So and these interactions will also depend on the, as I have discussed, as I have said many times, these interactions will also depend on the type of glycosylation that has occurred on the uh, surface receptors that are expressed on the immune cells and also on the antigen presenting cells. I hope you got a general idea how these lectin glycan interactions will impact the immune response. If you need to understand more about these lectin glycan interactions and its relationship with the immune response, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.